shall we?
Yeah, so it looks like everyone's trickling in. Good morning, everyone. Thank you so much for joining us today. I am so excited to introduce our very own Ben Liechty, who is an assistant professor of pathology who specializes in neuropathology here at Cornell. Ben decided to receive not one, but 2 bachelor's degrees from the University of Delaware, majoring in East Asian studies and then later in biology. He then went on to receive his MD at Thomas Jefferson University in Pennsylvania. Ben then, as many of you may not know, spent 2 years as a neurosurgery resident, but he soon saw the light and he shifted course and went on to complete his pathology residency at NYU, where he focused in anatomic pathology and neuropathology. He then completed a fellowship at Columbia in molecular genetic pathology, and he joined the faculty here in 2018. Among his many accomplishments, Ben is also a very talented programmer, and he's really been working to help us rethink how we use technology in pathology with a focus on machine learning algorithms. Ben has been a wonderful addition to our neurosurgery and neuro-oncology community here at Cornell, and we're so excited to hear about your work, Ben.
Right. Thanks so much, Rupa. I'm happy for the to talk about what we've been working on. So, let me just start with a quick overview. I just want to make sure everybody's on the same page with some basic machine learning terminology. We'll talk a little bit about some of the ways we can encode non-numerical data to then be used in interrogating a model. I'll talk a little bit about clustering, and I'll talk a little bit about decision trees and random forests, because those are applicable to the methylation array profiling classifier. We've talked about that several times. What I really want to focus on today is our work with convolutional neural network in computer vision. Sorry, can we mute whatever that sound is?
Oh, dude, he's on our paper.
Sorry about that. Okay. So just some basic machine learning terminology. One of the first questions you ask yourself is, do I have a gold standard in my data that I can train towards? Or do I want my algorithm to try to identify inherent structure within my data? If you have a gold standard that you can train towards, we call that supervised learning. If you don't have a gold standard, we call it unsupervised learning. There are also methods for dealing with sparsely annotated data by possibly imputing values or by masking out values which may have an entry in some objects, but not an entry in other objects, to try to overcome that limitation. In general, supervised methods require a lot of work on behalf of the data entry people, and unsupervised models require a lot of either computational power or a lot of work on behalf of the programmers. There's a lot of problems you can try to tackle with machine learning. So regression is when you try to map towards a numerical value. Say you have a model that you want to try to predict how long progression survival might be. Classification, if I want to predict what a given tumor class is. Clustering, which is if I want to try to group more like entities and separate out more dissimilar entities. And then anomaly detection. This would be trying to find things that don't quite fit a pattern. This is often used in things like identifying spam emails or whenever you get a call from your credit card company that there's been an abnormal charge. That's what anomaly detection usually revolves around. The great boogeyman of machine learning is this concept of overfitting. So overfitting is when a model performs overly well on the data that's been used to train it at the expense of how well it performs on real-world data. So there's a lot of possible causes for overfitting. One of the most common ones is you just don't have enough data to train it with. You may have a model that's too tightly constrained to your training data, or you may have training data that isn't really applicable to real-world data. So for example, suppose our lab is very, very well validated for generating neuropathology slides, but then we get slides from another lab that is better optimized for, say, breast cancer cases, which require a different type of fixation. They'll perform worse on brain tumor sections, and then our algorithms will, as a result, perform worse on brain tumor sections. In general, what you do to assess how badly overfit your model is you split your data into a set used for training and a set used for testing. So the training data is what's used to update your model. The test data is what's used to validate your model, to make sure that the weights that were learned from the training data are then applicable to real-world type data. So just a simple a demonstration of how this works. So this is a parabola I generated with just some random noise added in. The red is going to be used as our training data. The yellow is going to be used as our validation data. We can try to fit this to several different types of curves. So since this is supposed to be an order 2 polynomial, it fits best on the second order polynomial. Sorry, it fits nicely on the second order polynomial here. fits relatively well on the first order polynomial. If you do a very, very high order polynomial, an overly tightly constrained model, you can get a very good R squared value on this training data. But then when you go to look at how that performs on real world data, the underfit model actually performs much, much better than the overfit model. Obviously, we'll never do better than our exact model, our R squared model, our second order polynomial model. Again, if you don't have enough training data, you may get a very misleading answer for your model. So this is an example trained on just 3 data points on the left. On the right is suppose that all of our training data is constrained too tightly to one region, but the real world allows for a much broader range of possible answers. It'll eventually drift as you get further and further away from your range used in your training data. Whenever you work with data, you want to try to encode it into a way that can be used numerically. Because really, machine learning models tend to require some kind of numerical values to train towards. Numerical values are very easy to work with. If you have something that's, say, disease-free survival, some measured lab value, you may not need to encode that at all. It may benefit from some normalization within a small reference range, but not all algorithms require that. Binary values are usually pretty easy to work with. That would be things like, is this tumor IDH mutant versus IDH wild type? And it's very easy just to encode one of those as a 0 and one of those as a 1. For categorical data, and I'll show you how this works in a moment, we often use a method called one-hot encoding, where you create a vector of numbers where all of the elements that match your class of interest get encoded as a 1, and everything else gets encoded as a 0. Ordinal values, this would be things that have, say, like an integer ranked value. So, for example, WHO grade, right? There's no such thing as a WHO grade 1.5, right? And you can either do this with one-hot encoding, or you can just do it with imputing numerical or integer values to your data. There are other encoding schemes that exist for more abstract data types. I'll show you later on how we do this with image data. There are also ways to work with, say, word embeddings if you're doing natural language processing that I think are kind of beyond the scope of what we're going to talk about today. So this is how one-hot encoding works. So I'd like to start by showing you guys our friend Chewy. Chewy is going to be helping me out whenever I have an image that I need to give you a reference image for. So suppose we wanted to encode this image and have a labeled output for it, that output being the term dog. So what we would do is we would have this one-hot encoder of all of our possible values in our system. Everything that matches our value will get encoded as a 1. Everything else will get encoded as a 0. So that's our target for training with one-hot encoding. So once you've got your data encoded into a useful way, how do you train a model? Well, this is going to depend somewhat on what your model looks like. Some models will train on, say, or some models will produce an output just out of the gate. A lot of those will depend on things like linear algebra decompositions. But for things that are nonlinear, for things that require some degree of fitting, what you do is you pass your training data through your model. You see what the model predictions look like. You then compare your model's predictions to your gold standard. And then using some method, you try to then alter all the weights in your model so that your model performs better on your training data. So what does this look like when you actually do it iteratively? So this is from an early model that I trained for computer vision. And all these dots represent the performance on our training data. And this is an error function. The goal is to minimize an error function. So all our validation cases monotonically continue to decrease in error or in loss, while our accuracy tends to monotonically increase on our training data in accuracy. But if you look at what our validation data, our data that was used for making sure our algorithm is working correctly but was never used to update the weights, it bottoms out around generation 10 here. And then it begins to become more and more erratic. It begins to overfit, right? At the same point, our model starts to plateau with our validation data. Okay. So clustering. Clustering is a grouping of elements by high dimensional, by collapsing high dimensional samples by how similar they are. There's a couple of ways to do this. I'm going to talk mostly about dimensionality reduction here. Whenever you see, for example, those heat maps that have a dendrogram or a tree structure next to them, that's another type of clustering called hierarchical clustering. And in general, we like to collapse things down for human visualization into 2, sometimes 3 dimensions. So one of the more common and older and more well-traveled methods of doing this is principal component analysis. This uses linear algebra to collapse your data into the dimensions with the highest variance. and then it remaps all of your data in terms of those 2 or 3 axes. One of the more recent methods that's very popular these days is something called t-squared stochastic neighbor embedding. And this uses a probabilistic method to perform clustering in a nonlinear fashion. So all these images here on the right, these are hand-drawn digits, and each of these intensities within each of these pixels here represents a possible dimension. And if you do t-squared stochastic neighbor embedding on these images, you actually get very nice clusters. So, is my animation working here? There we go. So this is what t-SNE looks like when you train it. This is from just a sample data set that I developed using a high dimensional fractal that I then tried to collapse into 2 dimensions. The way this works is you initially lay out everything in a random Gaussian distribution, and then you compare every point to its neighboring points, and you can say how many neighbors you think each point should have. And if the points think they should be neighbors, they get moved closer together. If the points think they should not be neighbors, they get moved further apart, right? In general, t-SNE preserves locally complex topology better than PCA does. PCA wants to collapse everything into an ellipse for the most part, whereas t-SNE will often preserve any kind of complex local topology really, really nicely. It's a little bit easier to take a new data point and feed it through a PCA to see where it would land. The other limitation of t-SNE, it's very susceptible to variations in its initial random state. So if you use a different seed, you'll get a different random result every time, although your clusters should be relatively similar. So this is what happened when we did methylation array profiling and took the 10,000 most differentially methylated sites and fed them through a t-SNE clustering algorithm. So you can get a couple of insights through this. So tumors that derive from the same tissue of origin cluster together really, really nicely. So these are all from pituitary. These are all from pineal region. These are all meningeal. And here in red, these are all of ependymal derivation. So there's some degree of fingerprint that is preserved for the cell of origin that gets detected by methylation array profiling. In addition, methylation array profiling can pick up the fingerprint of different driver mutations fairly nicely as well. So here in yellow, we have all the tumors driven by IDH mutations. Here in blue, we have all tumors driven by INI1 or SMARCB1 loss. And here in purple, we have all the things driven by MAP kinase pathway alterations. For example, BRAF mutations or fusions or FGFR rearrangements. Now, some things clustered really nicely with whatever their tumor class was, and some things that have a histologically defined tumor class clustered very far apart. So, for example, glioblastomas or high-grade infiltrating gliomas, all the IDH wild-type ones clustered really tightly together here in green. Whereas medulloblastomas, we know there are different molecular subtypes of those that are very disparate. They behave very differently as well, and they clustered here in blue. So here's the sonic hedgehog ones. Here are the group 3s and group 4s, and here are the Wnt ones. So this shows that these are biologically very, very different. In addition, the IDH wild-type gliomas and the IDH mutant gliomas also clustered very, very far apart, supporting that argument that these are very, very different tumors biologically. So I just want to show you guys a little bit about how the methylation array profiling classifier actually works. We've talked about this a few times, so I don't want to belabor it too much. But the methylation array profiling classifier is built on what's called random forests. So random forests are sort of an evolution of the concept of decision trees. And decision trees are really easy for humans to understand. What you have is a root node here where I've decided I have an infiltrating glioma or whatever. I then pass it through branch by branch. Does this tumor have an IDH mutation, which would push me down this pathway? Does it have a 1p19q codeletion, in which case it would push me down this pathway? And then it gets me ultimately to what we call a leaf, and a leaf represents what diagnosis I end up calling it through this decision tree. The way you train a decision tree is you determine which dimension has the highest purity when you split it off at some given cutoff point. All right, and then you do the same thing with the split subgroups. The problem with decision trees is they're what we call a greedy algorithm. They may choose an early branch point, which is suboptimal, and it leads to more complicated branching downstream. So one way to solve this is by saying, all right, I'm going to make thousands and thousands of trees. And instead of me letting the algorithm decide what order the dimensions get split on, I'm going to say up front what order all those dimensions land on my tree. So for example, with methylation array profiling, once you enter at the root node, each of the branches represents a methylation site, a CpG island. And it can be either hypermethylated or hypomethylated. Then to determine the cutoff that you want to use for each of these branch points, you use what's called a bootstrapping or bagging algorithm. You pass your data through your classifier, you see how well or poorly it performs, And then you update all your weights to try to make all your trees predict the right class more often. So once this is done, you then have thousands and thousands of trees, and say you have 100 different tumor classes, but if 30 or 40% of your trees vote for a diagnosis, that really supports the argument that your case you fed into it represents that as opposed to just a random chance. It's kind of similar to if you take a case that's problematic and you show it to 100 pathologists. If, say, 40 or 50 of your pathologists all agree on one diagnosis and all the other pathologists are scattered across a wide variety of diagnoses, it kind of lends argument to the idea that you should manage it as if it was whatever the majority of people think. So the percentage of trees voting for a given entity is referred to as the raw score. What you can then do is you can then compare from your test cases your sensitivity and specificity, and you can find out what the threshold value is, and you can remap your raw score into what's called a calibrated score. So for methylation array profiling, if you have a methylation score greater than calibrated score greater than 0.958, they were calling that a match. Anything under that means it's maybe something that's not in the classifier, maybe some novel entity, it's maybe a technical failure, but a no match is not really considered to be an informative result if it doesn't pass any calibrated score. This is what it looks like when you take those calibrated scores and then remap them, those raw scores, and then map them into correlated scores. So let's move on to neural networks now. This is what I really want to focus on for the most part today. So neural networks are a type of machine learning algorithm that focuses on converting tensors, which is essentially an organized box of numbers of a fixed size and shape, into a different type of tensor of a fixed size and shape. Neural networks have gotten really popular in the last few years because they're able to perform very complex tasks that usually require a human to do. So things that are very, very abstract, like image recognition, like speech recognition, speech generation. So a lot of movie reviews now are actually written by neural networks, at least in part, and then just reviewed by a human. So with sufficient training data, neural networks can hypothetically perform better than humans in some circumstances. It depends on what your gold standard is. If your gold standard is a human annotation, you will never beat the human annotation. So the idea behind neural networks was really inspired by the layered representation of how we process data in neurons in our brain. That's how they got their name. So what kind of things can be encoded into tensor? So a zero-dimensional tensor is just a number. That might be your progression-free survival. A vector would be a one-dimensional tensor. That would be, for example, one of our one-hot encoding vectors. A matrix would be a two-dimensional vector. That might be, say, a grayscale image. Higher dimensional orders are tensors are referred to by their rank, by however many dimensions they have. So one-dimensional tensors, text, sound data, things like that. Two-dimensional tensors could be grayscale images. Three-dimensional tensors. So if we take an image, right, it has a height and a width, and then it has a depth for a color channel. So the red, green, and blue color channels can be thought of as a third dimension to our image data. Right? Volumetric data from, say, some radiographic imaging can also be thought of as a third type of dimension. Four-dimensional tensors. So suppose you had something that had length times width times color channels times time. That would be video data. You can work with video data using tensors. So a simple schematic, I'm sure you guys have seen things like this before. This is what neural networks tend to look like. You have these input data points, and these could be anything that's of a numeric value, lab values, intensity of pixels, complex embeddings derived from some other machine learning algorithm. You then pass those through a layer of weights, you multiply them by some number, and you add them all up and you put them in some hidden layer here. And then within the hidden layers, you do some nonlinear transformation. You lose the benefit of layered representations if you just do linear transformations. You can get much, much more complicated representations of your data if you do nonlinear transformations. And there are many nonlinear transformations that can be used. It depends on what you're trying to do with a given layer as to which one you might use. Some of them are useful for certain types of outputs, but I don't want to get too much into that right now. If anybody wants to talk about that, feel free to check in with me personally later. So image data is encoded by three-dimensional tensors. So the height and width channels represent just the intensity of a pixel at a given X and Y location on your image. And then the third dimension represents the red, green, and blue color channels. Let me introduce you to the idea of convolution. So convolution is how we do most of our work with computer vision these days. It's really the kind of the modern standard for doing computer vision. If you try to have an algorithm that just allows for every single point in an image to talk to one of your dense layers, you get a very, very large number of parameters very, very quickly. So that becomes intractable. You can't get that into your computer's memory to actually train your model on. It's very computationally expensive to do that. And something that's a little bit less obvious. If you have an image that is the same image, it's been translated, it's been moved over from one position to another, an algorithm that doesn't understand there's local topology, there's local meaning to data, that points next to each other have some relevance to each other. You'll never be able to learn the idea that Chewy in the bottom corner is the same as Chewy in the top corner. So to get around this, we use the idea of convolution. So what is convolution? Convolution is a way of passing a filter over our image and then redrawing our image in a more abstract way, okay? So let's start with a simple example with this tic-tac-toe board. Suppose I wanted to find everywhere where there was an X on this tic-tac-toe board, right? What I would do is I would then pass this filter over my image. Let's see, there we go. And the better my little filter here, the better my little area of interest here matches my filter, the higher a score I'm going to give it here. Okay, so I'm going to pass through. This matches in about 7 out of the 9 pixels. The next one's much, much worse. This matches, I think, about 4 out of the 9 pixels. Sorry, 1 out of the 9 pixels. This one matches okay again. And we go on so forth down the line. And we end up getting this heat map after we've done this activation map. And everywhere where my filter matches perfectly, we get a score of 1. Everywhere where my filter matches terribly, we get a much lower score. And we can do this for multiple different types of filters, right? So suppose I wanted to have an O filter. Here it's going to pick up all where my O's lived. If I wanted to assess the cross-hatching here, right, I could have a plus filter here that gets lit up by the cross-hatches. I could identify the horizontal lines. I could identify the vertical lines. So these filters are what I want to try to get my model to learn. All right, so let's combine the idea of convolution, the idea of making these filters, with the idea of layered representations. That's what we mean by convolutional neural networks, right? So we have block after block after block of convolution, then passing this data that they've learned from their filters to the next layer. And our goal is to optimize those filters so that they match so that they give us good output data, right? So what does that look like in the real world? So this is a simple classifier that I'm going to show you in a little bit that predicts different anatomic sites from autopsy cases. This is a high power shot of cerebellum with a large Purkinje neuron here, with some granular neurons here, okay? This is our earliest layers, middle layers, deeper layers. There was several much deeper layers in this that had so many tiles in I couldn't even fit them on the page here, so I'm not going to show you those. But I will show you some selected ones. So these are from the earliest layers. Early layers detect things like edges, like white space, like solid areas of purple, right? Middle layers start to learn more abstract features. So here we have just a detector for the cytoplasmic membrane, the rim of all of our Purkinje neurons, right? This one here detects all of the cytoplasm of our Purkinje neurons. This one here detects the nucleus of our Purkinje neurons and our other neurons, as well as the Nissl substance within our Purkinje neurons. As you go deeper and deeper, you get more and abstract things. So here, this one picks up this dendrite here. This one here seems to only be really picking up for our granular neurons and is ignoring all the other cells in the field. As you get to the deepest layers, you start to get things that are really salient to classifying your image. So this picked up a combination of the Purkinje neurons, nucleus, Nissl substance, and these granular neurons. But they get very, very abstract as you get deeper and deeper. It's harder to understand what they're actually detecting. So let me tell you about this toy data set that we developed when we were trying to get all this working in the first place. So while we were developing our code base, I wanted to have a sample set of images to work with that had the following characteristics. I wanted something where all of my classes looked like the same in every single case. So one of the problems we run into with gliomas are very heterogeneous. We often refer to glioblastomas as the gift that keeps on giving morphologically. I've seen gliomas look like kind of everything under the sun. But every single cerebellum from every single autopsy will look like every other cerebellum from every other autopsy, right? A relatively large N for each class. So we can get lots and lots of tiles out of our autopsy sections. I wanted to have images that a trained human observer could easily classify just by visually alone. Something that I could teach a 5-year-old child to identify with maybe a half an hour worth of work. So this is why I chose to do autopsy slides as my toy data set. I wanted things that we'd sample in every single case. So I selected 11 regions of interest that I thought were easy to identify. I took good representative images that I thought would be very, very easy to pick up and that has some pathognomonic feature for that region of interest at 4, 10, 20, and 40x objectives, optical objectives on my scope. I generated a total of 872 source images. There were a few cases where I didn't have a good section of some particular region, so we ignored those. If I didn't have a good Luxel fast blue section on something, I required a recut. I would use an H&E for those sections. So these are the areas of interest. I wanted things that would be somewhat confounding. So watershed cortex and occipital cortex look very, very similar, right? They're still 6-layered neocortex. Sometimes you can appreciate this faint blue line of Juthani coming in the occipital cortex. I wanted some things that would be a combination of gray and white matter, right? Because these are Luxel fast blue stains. So striate nuclei always have these thick bundles of white matter running through them. We call these the pencillary fibers of Wilson. Whereas thalamus is a mix of gray and white matter, but the white matter is a little bit more interspersed, more vaguely infiltrating through the gray matter. I wanted some things that were a combination that had a very characteristic pattern. So pons has crisscrossing white matter fibers going through the plane of the slide and in and out through the z-axis of the slide. And it's intermixed with these pontine nuclei. The medulla oblongata has this tortuous serpiginous nucleus here. This is the inferior olivary nucleus. And then ventral horn of spinal cord will have this combination of this butterfly-shaped rim of gray matter with large anterior horn neurons, and then a rim of white matter tracks. And then some things that nothing else would look like. Substantia nigra will have these pigmented neurons. Cerebellar cortex, nothing else looks like that. Nothing else has the same kind of complex architecture as that. Anterior pituitary is the only thing in my set that looks epithelial at all. And hippocampus, you can appreciate very easily has this nice dentate line in it. So things that would be very, very easy to pick up for a human. I took these images at 4, 10, 20, and 40x objectives in the idea being that certain things would be easier to detect at high power, certain things would be easier to detect at low power if you had either a cytologic versus an architectural feature that was useful for classifying it. So let me introduce you to the idea of augmentation now. So augmentation is taking the data that you have and making the most out of it. So I don't want my algorithm just to memorize my source images, right? I want it to be able to identify those images even if they're moved around slightly, right? Because a human can identify very, very easily that this translation doesn't change the fact that this is the same picture of the same dog. Scaling this image, it's obvious to a human observer. This is not a different animal. This is not a different image even, right? Rotating. I wanted to make sure that my algorithm could predict things if I rotated them slightly. And then you can also do color channel adjustments. I didn't do that for this. I will show you that when we do our whole slide scan processing. So I took our images. They're very, very large. I tiled them to 256 by 256 pixels. I repeated this process justified to all 4 corners to give me some translational modification. I then shrank the images down, performed the same tiling process. I then augmented it further by mirroring the tiles and then doing 90, 80, and 270 degree rotations of all these to multiply my number of tiles in by another 8, right? Once I did this, from my source images, I had about 2 million tiles. So this is the justification to all 4 corners. This is the downsampling where I shrank my images down. This is the mirroring and rotations, right? I then split the cases so that no case was in both the training and validation sets. I took 14 cases and I took all the tiles from those cases and used them for training. I took 5 cases, split them entirely for validation. And there's one case where everything was particularly well preserved. I set that case aside and I used that for post hoc analysis after my model was trained, right? The first version I did was a simple model that I showed you earlier. So here's our convolutional base where we remap it into these abstract filters. Then I collapse them down into what we call dense layers. Once I had them small enough, I take these dense layers and then you combine those into your final classifier. Okay. This is the training curves I showed you earlier. This is what it looks like when you track the error function on the training set versus the validation set, the accuracy on the training set versus the validation set. I ended up getting, with this first model, a peak tile level accuracy of 54%, which is not great, far better than random chance, all right? So accurate calls, I think it's useful to see what it gets right and what it gets wrong. all the accurate calls had some feature that I thought was straightforward for me to identify what I was dealing with here, right? So I see these pencillated fibers of Wilson, I see pigmented neurons, I see crisscrossing white matter fibers, right? I see something that looks very, very glandular. I see something that looks like granular and Purkinje neurons. I see dentate line. These are all things that I could use very easily to classify these. The things that got missed, right? They tend to be things where it's a very, very zoomed in image, where it's all just a field of gray matter, right? A field of just white matter. And then things that are zoomed in or have some confounding features. So for example, this is supposed to be from spinal cord, but it saw this large anterior horn cell and it thought this was a Purkinje neuron. So it called this with high confidence the cerebellum, wrong class. One of the ways you interrogate how well your model is doing is with a confusion matrix. So confusion matrix maps my target classes, my gold standard, versus what my predictions were. What you want to see is you want to see a nice staircase going up the side here. Everywhere where there's deviation from the staircase tells you there was a problem where you had a misclassification. Now, a lot of things that got misclassified, things that had a lot of white matter tend to get misclassified as other things that had a lot of white matter. Things that had a lot of gray matter tend to get misclassified as things that had a lot of other gray matter. In general, things did a little bit better as you went to lower levels of magnification. So we really had a tipping point at intermediate levels of zoom. I'll show you that when I had this working a little bit better with one of my upcoming slides with one of the later models I trained. These are the softmax predictions for the target class by objective and by anatomic site. So what you want to see here, the better your model did, the more of a flat line you'll have at the top here, where it was more certain that it was dealing with the correct class. So for some things, watershed was a lot of gray matter, and occipital was a lot of gray matter. As you get to higher levels of magnification, your error really goes up a lot on these, right? But for occipital, because it has this pathognomonic feature of this blue line running through it, as you go to lower magnification, you do much better with occipital. It always had a great job with pituitary. It tended to do a pretty good job with substantia nigra. It tended to do a pretty good job with hippocampus. Tended to do a pretty good job with cerebellum. So I think we can do better than 54% though. So there are some strategies that we can use to improve the performance of our model. So regularization, we could penalize our model for trying to be too complicated. We can do something called dropout. Dropout is where we randomly select nodes to be ignored during training for every single step of training to prevent them from conspiring with each other, to overfitting to a given class, right? From finding some noise to train towards. We can take our most accurate model and then stop training it once we stop improving. We call it early stopping. We could also try taking the we could also try taking a network that has been trained on something else that is potentially transferable to the data we're using now. So let me introduce you to the idea of transfer learning. What happened here? There we go. Transfer learning. So transfer learning is taking a network that's trained on one type of data. So for example, if some horrible hipster took pictures of everything he ate at restaurants over the last year, trained a classifier to predict what they were, and then took the classifier, cleaved off the output layer, and then took these weights from this trained classifier, and then tried to train them on, say, all the desserts they'd eaten in the last year, and then make new predictions from that. And it turns out this tends to improve your accuracy as long as your original data looks enough like your new data that you're trying to train towards to derive some information out of it. The features are somehow transferable. So there's no good trained model out there for histology data, unfortunately. But there are well-trained models out there for vision in general. So there's this data set existing called the ImageNet dataset. It's continually growing over the last 10, 15 years. The current version has about 14 million images across 21,000 different classes. And these will be pictures of animals, these will be pictures of scenes from nature, these will be pictures of kind of anything under the sun that you could potentially stick a name to. And it's used to benchmark convolutional neural networks. So there's some commonly used ones that have names like Inception V1, 2, and 3, which were developed by Google, VGG 16 and 19, which were developed by Google, Exception, which was developed by another ex-Googler. So the question I asked myself was, well, are the features from these pictures of nature, are these transferable to histology data? So this is just kind of what the architecture of Inception looks like. It's a little bit more complicated than my simple model. It has these branching structures which allow it to identify things at different levels of size. So to test whether transfer learning was useful from these ImageNet datasets, we took 3 commonly used neural networks, Inception v3, VGG19, and Exception. And we trained our dataset on these, okay? So we took the entire model, took the weights from those, and allowed the entire model to fine tune on our autopsy images. We also then took a version of the model that was randomly initialized. That's how you typically initialize these, with no starting weights. It's just random noise. And then you train from there, all right? So we did 5 versions of each of these models to demonstrate how reproducible they were. And then we would stop early whenever we had the best version of a given model. And this is what it looks like. So all of our pre-trained models, this is Inception v3, this is VGG19, this is Exception. All of these, when they're pre-trained on ImageNet, do much, much better. They're closer to 70% accuracy compared with our uninitialized or our randomly initialized versions of the model, all of which really peak out between 40 and 50%. This is the confusion matrix between my best performing pre-trained model, which was one of my Inception v3 models, and the confusion matrix from one of the uninitialized models. This is one of the best initialized, uninitialized model. So the staircase here is much, much better. The deviation from this is much, much better than it is in the randomly initialized version of this model. So something interesting you can do, if you want to get some insight into some of the layers of your model, but they're not amenable to the feature mapping, the activation mapping I showed you earlier. To get some idea of what our dense layers learned, are layers that don't have any kind of topological information in them. You could take all the images that you run through that model, and then you can take those weights from those intermediate layers, and then you can do clustering of them. So this is a stochastic neighbor embedding from the second to last layer from my top performing ImageNet initialized model here. All the translucent dots are things that it got correct. All the dots that are filled in fully are things that it got wrong. And in general, it kind of makes sense the things that it gets wrong, right? It has a hard time telling watershed cortex from occipital cortex, right? It has a harder time telling medulla and other like white matter structures from cord, right? This is the same thing done with my top performing randomly initialized model. The first thing I'll point out to you is there are many, many more points that were miscalled here, right? There are many more points that are filled in here. Our clusters are far less better defined here as well, reflecting the fact that this model had a much harder time telling things apart, right? One of the other insights that we gained from this are images that were taken at intermediate levels of zoom here, right? So this is all the way zoomed in at the highest magnification. This is all the way zoomed out. Our best performance tended to be at more intermediate ranges of zoom. So we've been using that as we move forward while we work with whole slide scans on tumor data. Okay, so now that we have that working a little bit more, I'd like to show you what we're doing right now. And this is very much work in progress. We started working with whole slide scans a few months back. Right, whole slide scans are very useful because that's how we would generate our data if we're going to try to do something with clinical data, right? It's not really practical for us to go and annotate a region of interest for every single case. It would make a lot more sense if we just could do this somehow automatically, right? So there are some problems with working with whole slide scans though, right? You have to deal with white space, right? There's a lot of white space you have to find a way to remove. Right? A pathognomonic feature may only be present in one area of a slide, right? You may have large areas that are all just blood, large areas that are all just normal brain, large areas that are just cautery artifact. So the question of how to work with that, that's something that we're still working on now. I'll show you some of our strategies to deal with that. So whole slide scans are arranged in pyramidal layers. And what we can do is to get rid of the white space, we can tile at the lowest level of magnification. And anything that's not informative, we can just throw those out. But anything that isn't potentially informative, has some tissue of interest in it, we can then tile those further, and then throw out the ones of those that are uninformative. And we can repeat this process until we get down to the lowest level of magnification. We can augment those tiles the same we did before. These are all the same region, just with different color channel alterations, different translational transformations, different rotational transformations, and different mirroring transformations, right? Segmentation. So segmentation is what we call it when you take human level annotations, right? Say if some expert were to go and annotate the regions of the slide they thought were normal brain, were reactive brain, were blood, were tumor, were palisading necrosis, were microvascular proliferation. And we take those and we try to redraw those as a set of tensors, right? And these could be our tensors that we train towards. Now this is fairly labor intensive. I will show you kind of a work in progress version of this in a moment. We're still working on trying to streamline this. Our data set for this is still relatively small, but ever growing. We do have about 600 scans as of right now. We only have about half a dozen that are really deeply annotated right now. We did do a pass through with trying to take the whole slides without trying to do any of the segmentation and predict the presence of IDH mutation. So this was a three-class classifier, which was trained to predict either IDH mutant astrocytoma, oligodendroglioma, or IDH wild-type astrocytoma, right? And the more yellow it is, the more it thinks it is that class. So I passed this filter over the entire whole slide scan. This is in fact an IDH wild-type astrocytoma. So it did an okay job with this one. I will say this model didn't perform as well as one might hope when you take a closer look at it. It does assume that far too many things are IDH wild types because it was weighted with just the raw number of cases. And IDH wild types only perform about only comprise about 20 to 25% of our data set. So when we tried to do this by weighting this so that they were all even, it does slightly better than random chance. It gets about 40% accuracy when you expect it would be 33%, which is still not great, but again, better than random chance, and that's a work in progress. So the other thing that we're doing right now, I would still consider to be still a work in progress. These are what happens when you try to take those segmentation tiles, those tiles that were manually annotated, and then number one, try to redraw the source image. Oh, this is what's called an autoencoder. Autoencoders are very, very notoriously very, very blurry, what we call very lossy. And then it tends to overcall things like solid tumor, and it tends to overcall what we call region of interest. Basically, at the moment, this is just a fancy white space detector. Like I said, we've only been working on this for the last month or so. I'm trying to get this working a little bit more functionally. So that's all I have to show you for right now. These are some references. And if anybody has any questions, I'm happy to field. Oh, I should also, where'd my acknowledgements go? I'd like to acknowledge David. I'd like to acknowledge our med student Cheyenne, who's been a real hard worker with this, been instrumental in curating our cases. She's been helping us develop our database to put together all the mutations with our tumors. I'd also like to thank our collaborators up at Ithaca campus from the School of Engineering, Mert Bunku, Zhulu Zhang, and Leo Moon. That's it. Oops.
Thanks so much, Ben. That was great, and I think an excellent introduction to machine learning, and hopefully everyone has some ideas about what they can do with that. I'm really interested to hear from you what you think the future is here and how close we are to it. So, specifically, how close are we to some of these algorithms replacing people, and where do you see replacement versus augmentation? And then specifically with regard to subspecialties, are we performing now at a level where this is better than a general pathologist reading neuropathology? Or, is that sort of the level we're at? And how long do you think it will take to really replace as opposed to just augment?
Sure, of course. Well, obviously, we're not the first people to come up with this idea. The group at Page AI actually developed a classifier for predicting areas of prostate cancer. And they took this with the data generated over at MSK. And they compared it with how human observers do. So what they ended up finding was, you can train a model to predict prostate cancer that does perform a general pathologist, but they could not outperform the GU subspecialist trained pathologists. So that's where we live right now. As far as how Neuropath is going, I will tell you that David and I did an under the table investigation for how well a human can do predicting IDH mutation. Just so you guys know, a human can do about 75 to 80% accurate predicting IDH mutation from H&E stain alone, which I was pretty happy to find that. So we're still outperforming the algorithm by a pretty good clip here. The features that you look for in gliomas are very different than the features you look for in infiltrating carcinomas, right? Carcinomas are gonna make still glands or nests of cells. And there's more architectural information that you can work with from those. Whereas gliomas, they may be very, very subtle, especially the IDH mutant ones, right? They may just be mildly cellular, and it may depend on just doing an IHC to pick up the IDH mutation, right? As far as where the future goes, my personal belief is, we've seen this kind of thing come and go for the last 30, 40 years, right? Back in the 80s, everyone thought immunohistochemistry was going to replace everybody's jobs. And no, it didn't. we all learned to use immunohistochemistry, and we got much, much better at our jobs, right? Back in the 90s, flow cytometry was becoming a thing. Everyone was like, oh, no one will ever look at a heme path flight ever again. And I have one of my residents here is going into heme path. I'm sure he can agree with me. No heme path person in this earth would sign out a case from the flow alone and give like a really definitive diagnosis. You really need histologic information as well. Molecular, right? Everyone said, oh, no, you're not going to need to do any of this histopathology nonsense ever again. We're going to classify these things entirely based on their methylation profile, on whatever driver mutations we identify in them. And as Rupa can confirm, we've had some cases that had things that could easily be passed off as high-grade tumors, but because I had some histological insight into how they looked, that we were a little bit more judicious about. And so far, so good with most of those. My real vision is that this is going to require both a human to be a sanity check on this, right? This will work well for, hey, did you notice this area? Oh yeah, I didn't notice that. Thanks for boosting me up another 5% accuracy. Or it'll be really useful for, say, secondary review of cases as, again, a sanity check. I think we're not that far off from us having this enter the clinical setting, but I think we're a long way off from replacing humans. I think machines make stupid mistakes that a human would never, ever, ever make. Machines are very susceptible to adversarial things. They're very susceptible to things they've never seen before, So that's my take on that.
Great, thank you. Does anyone else have any questions?
Ben, I have a question. Jonathan Nisley here.
Please.
Ben, the tumor microenvironment is obviously of interest in terms of the host reaction to the tumor, as well as the impact of any interventions that may be taken on the tumor, and whether there has been any work that you're aware of that has begun to try to interrogate the tumor microenvironment using the techniques you've described today.
Sure, that's an interesting question. Not that I'm aware of. Now, what you could do, right? We could have the immunostain of interest be one of our input labels, right? We could have, you can kind of put together a combination of, say, a convolutional neural network with some other classifier, and then join those together, and then you can have multiple output classes from those as well. So what you could do is you could feed an algorithm multiple tiles of, say, different immunostains, and in addition to each of those immunostains have a label for what specific marker they're picking up. One could train a network to do that. I'm not aware that anybody has actually done that yet. It would be conceivable to do. It would be a little bit laborious. I'd want to make sure that the rest of our network stuff is working before we try to do that.
Hey, Ben, great talk. This is Dave. I just had a comment to sort of follow up on some of Rupa's thoughts and my own thoughts in terms of where we are. And I think, one of the things that's important to think about is a lot of the algorithms that are developed are very task dependent. I mean, they're very specific to a particular task.
Yes.
So I think one of the things that's underestimated is the degree of generalizability of some of these things to, the larger picture, and that relates to, the full realm of a person's job, right? So like replacing a whole neuropathologist is different from saying the particular task of predicting IDH mutation, right? Those are totally two different things. And so, just the same way as, when Tesla's, sort of trying to figure out how to drive, it's one thing to say, is the traffic light red or green? And it's another thing to do completely fully autonomous driving in the city.
Yeah, for sure.
And so if you think about it in those terms, one of the barriers, in fact, I think, to pathology and diagnostics and using this stuff is that we don't have systematically encoded outcomes data.
No.
And so really what pathology is trying to predict and guide with respect to an expected outcome. And so if you treat the outcome itself as the gold standard for whatever your diagnostic rubric is, that's in fact the data that we're missing. We don't have that data. And so until we're able to access those sort of outcomes data in a more systematic and computational way, I think it's going to be a long way off from these types of things replacing humans.
And David and Ben, that was actually another question I had. Ben, you had mentioned that there are no shared algorithms that have been developed in pathology or neuropathology. But what about shared input data? You know, that seems like the quality of these algorithms is completely dependent on the amount and quality of the data. Are there like curated high quality data sets like TCJ or, you know, places that you can pull from to help validate?
Sure. I mean, TCJ is certainly a good resource, and our engineers are working with the TCJ data right now. There are some other databases out there for normal histology. If anybody's super interested, I can send you the links to those. But yeah, nobody's really sat down and tried to develop models for these that have been publicly released yet. Everybody's still holding onto those fairly tightly at the moment. Intellectual property is valuable.
Okay, thank you. Well, thank you so much for joining. I think it's been really interesting, and please feel free to email Ben if you have any questions or want to talk offline.
Thanks so much again for the invite, Rupa.
Thank you. Thank you, everyone.